Assalamualaikum and good day everyone. Today I would like to give you an introduction to the first topic of this course, Dengue and Zika, a global health threat. By end of this topic, students should be able to know the basic biology of dengue and Zika viruses, the world distribution of both dengue and Zika diseases, how dengue and Zika are transmitted to humans, and why both dengue and Zika pose a significant threat to the community. Before we go further, I would like to begin by introducing you to some facts on dengue and Zika. Dengue, as we know, is one of the most important mosquito-borne viral illness, which is caused by infection with dengue virus. The virus has 4 different serotypes, which are dengue virus 1, dengue virus 2, dengue virus 3, and dengue virus 4. All these 4 dengue virus serotypes share approximately 65% of genome homology with some genetic variations within a single serotype. Infection with one serotype confers a long life immunity. However, a secondary infection with a different dengue virus serotype does not confer any protection. This could lead to antibody-dependent enhancement, or ADE, which causes a high risk of severe dengue complications. Dengue is typically self-limiting with a mortality rate of less than 1%. Clinical manifestation of dengue varies from virtually asymptomatic cases to catastrophic situations with bleeding and multiple organ failure. Most dengue patients experience clinical symptoms beginning day 3 to 14 of infection, which include high fever, retroorbital headache, muscle and joint pain, nausea, and skin rash. In a small number of cases, the disease may develop into severe life-threatening forms, which are dengue hemorrhagic fever and dengue shock syndrome. This results in bleeding, low level of blood platelets, and blood plasma leakage. Another important mosquito-borne disease is Zika, which is caused by infection with Zika virus. The virus was first identified in monkeys in Uganda back in the year 1947 and was later discovered in humans in 1952. Symptoms of Zika are almost similar to dengue. However, unlike dengue, Zika may cause congenital brain abnormalities, including microcephaly in infants.
The Zika virus disease is caused by a virus transmitted mainly by mosquitoes of the genus Aedes. Subjects may develop moderate fever, rash, exanthema, conjunctivitis, muscle and joint pain, discomfort or headache. The Zika virus took its toll in 2016, wherein thousands of mothers and their babies were highly affected by the disease derived from mosquitoes. The Centers for Disease Control (CDC) then announced that mothers who were struck by Zika are 20 times more prone to have children with birth defects.
Just with each test, one by one, it gives us more and more of a better idea of what direction to go in.
We are working on something that's a little bigger than what we are. This is moving at an unprecedented speed. Right? It's moving quite quickly. We manufacture a vaccine within 6 months, from very beginning to the very end.
We use past experiences with other viruses to try to relate to Zika to better understand it.
The vaccine has already been completed. we're testing the vaccine as well. Once it's given to that person, all of those samples come back to us and we tell them, yes, it works. It creates neutralizing antibodies. It's what we want. Our lab has been determined to be one of the testing labs for almost all the human clinical trials. So the soldiers that we have in our lab will be part of that. One of the things that I've always done is I've always asked for soldiers. Because I think when it comes to the soldiers, you get to teach them something that they're probably not going to encounter being a soldier.
I actually wanted to go to a field unit and got stationed here, and it's turned out to be a wonderful experience for me. I feel really lucky, really lucked out.
Making sure the vaccine works well, that all these tests that say yes, the vaccine is ready to go inside a human person for testing. Once all of that is completed, and hopefully by September of this year, that will be done, then clinical trials will start moving ahead.
Microcephaly is a birth defect where a baby head is smaller than expected when compared to babies of the same sex and age. Babies with microcephaly often have smaller brains that might not have developed properly. Severe microcephaly is a more serious, extreme form of this condition where a baby's head is much smaller, which has not developed properly during pregnancy or damaged at some point during pregnancy. Depending on the severity of infection, babies with microcephaly may encounter several problems such as seizures, hearing and vision loss, development delay, and intellectual disability. So as we know now, dengue and Zika is a disease of significant importance and has become an international public health concern. With more than 1/3 of the world's population living in areas at risk for infection, both viral diseases may cause illness and death, particularly in the tropics and subtropics. Both diseases are endemic in much of Asia, including India and Southeast Asia, as well as Central America and also Brazil. In recent decades, the incidence of dengue has grown dramatically around the world. The actual numbers of dengue cases are underreported and many cases are misclassified. One recent estimate indicates about 390 million of dengue infections per year, of which 96 million manifest clinically. In another study of dengue prevalence, about 3.9 billion people of 128 countries are estimated to be at risk with dengue virus infection. This is a worrying scenario as an increasing trend of the disease are seen in very recent years.
Would you be able to describe in general the trend of dengue cases in Malaysia?
Okay, there was a big outbreak of dengue starting from the end of 2015 until early 2016, almost throughout Malaysia. However, I would say that the trend of dengue cases this year is in a decreasing trend. Until epic week 46 or until 18 November this year, 2017, there is a total of 78,062 cases compared to 93,080 cases during the same period in 2016. So there is a reduction of 16.1%.
Dengue and Zika viruses belong to the genus Flavivirus of the family Flaviviridae. Do you know how do these viruses spread? Both viruses are transmitted to humans through the bite of an infected mosquito. only a few mosquito species are vectors for dengue and Zika viruses. These are mosquitoes of the genus Aedes, particularly Aedes aegypti and Aedes albopictus. Through these mosquitoes, dengue and Zika viruses are spread by a human to mosquito to human cycle of transmission. Typically, after being bitten by an infected Aedes mosquito, a person will develop viremia, a condition in which there is a high level of virus in the blood. Viremia lasts for approximately 5 days, but it can also last as long as 12 days. Within this viremic period, a mosquito may feed on the blood of the infected individual, and the virus will eventually spread through the mosquito's body and saliva. So this mosquito can now transmit the virus to another healthy person while taking a blood feed, and this cycle of viral transmission goes on. Apart from knowing the disease transmission cycle, it is also very important to understand the process of viral infection in mosquito. Initial infection occurs in the epithelial cells of the mosquito midgut, spreads through the hemolymph to other organs of the mosquito, such as the fat body and trachea, and finally disseminates to the mosquito salivary glands. Once in the saliva, dengue virus can be transmitted to another susceptible host when the mosquito acquires a blood meal, thus spreading the disease. Female mosquitoes are responsible for carrying these viruses and cause infection in humans, as they require certain proteins in the human blood to develop their eggs. So do you know why dengue and Zika pose a significant threat? Well, transmission of dengue and Zika has significantly increased in urban and semi-urban areas across the world. And this is facilitated by the highly efficient modern transportation and Aedes mosquito vector transmitting the virus, resulting in a local spread within a community or to a different geographical region. Further, the existence of 4 dengue virus serotypes complicates treatment of dengue and hampers the development of an effective vaccine. In endemic areas, limited sensitivity of early diagnostic tools and the cross-reactivity of antibodies to previous infections may lead to improper care and increase the risk of death. And this is also worrisome because clinical features of mild dengue and Zika fever sometimes resembles clinical symptoms of other viral infections, which leads to misdiagnosis of and can lead to mortality in patients. Treatment of patients, environmental management of the mosquito vectors, and disease prevention program have imposed a massive economic burden for the governments and communities. Taken together, dengue and Zika pose a very significant threat and economic burden to many countries.
Is there any dengue cases reported in UMT this year?
Alhamdulillah, there is no dengue cases reported within UMT campus this year. However, there is one case
reported in Kampung Tanjung Gelam involving our out-campus student.
What are the preventive measures or actions taken by UMT in order to prevent dengue transmission?
First, we had recently organized a program called Gempuk Edis UMT on 28th October this year. This event was co-organized with our local authority, the Department of Health Negeri Terengganu, and highly participated by UMT students, as well as the local residents of Kampung Kumang Badak. This program involved gotong royong, or we call it community spring cleaning activity, to destroy the breeding sites of Aedes mosquito in Kolej Siswa and the nearby residential area. Other mosquito control operations have been accomplished to reduce mosquito population and destroy their breeding sites, which include: we established a Kombi UMT committee, which consists of representative of each school in UMT. Then they will be trained to perform routine self-checks within the school every week. Okay, for your information, Kombi stands for Behavior Impact Communication. This program, founded by the WHO or World Health Organization and implemented by the Ministry of Health. Kombi is a dynamic approach that uses social mobilization and communication strategy to influence behavior change among individuals, families, and communities towards healthy behaviors. We also do the mosquito fogging operation had been conducted within our University Kolej Siswa in February and September this year. This activity also included oiling, which were sprayed into the drainage system to eradicate mosquito larvae. Others, we also did the gotong royong or community spring cleaning within all schools and administration building from February to April this year. We also create public awareness and promote health education on dengue and its mosquito vector campaign by producing pamphlets and brochures. We also disseminate larvicides to public for free to treat mosquito larvae at home and keep home free of mosquito. Close monitoring by the University Health Centre throughout UMT campus. We also increase health education and public awareness through several ways. For example, circulation email to all university staffs and students, social media through University Health Centre Facebook and Student Representative Council, and also WhatsApp's application, and also we open a booth from University Health Centre during graduation ceremony.
I would like to discuss about the virion structure of dengue and Zika viruses. Similar to other flaviviruses, both dengue and Zika viruses have a nucleocapsid core containing the viral genome and multiple copies of capsid proteins. This is surrounded by an envelope derived from the host lipid bilayer. Fitting crystal structures into cryo-electron microscopy map reveal about 180 copies of the envelope glycoproteins organized as 90 dimers in a herringbone pattern on the surface of the lipid bilayer. Similarly, 180 copies of the membrane protein are present on the surface, partly integrating with E proteins. So part of the E and M proteins are anchored to the virus lipid bilayer, exposing the alpha helicus of the virus envelope. The following picture shows the dengue and Zika viruses. Both dengue and Zika viruses have a positive sense single-stranded RNA genome of about 11 kb in length. And this is flanked by highly structured 5' and 3' untranslated regions. Similar to other flaviviruses, the RNA is capped at the 5' end and lacks a poly-A tail at the 3' end. The genome encodes a single open reading frame, which is then translated into a large polyprotein precursor consisting of 3 structural proteins: capsid, premembrane, envelope, as well as 7 non-structural proteins: NS1, NS2A, NS2B, NS3, NS4A, NS4B, and NS5. Dengue and Zika viruses replicates in host cells. The virus initiates an infection by entering the host cell through receptor-mediated endocytosis. Upon entry, the viral E glycoproteins undergo trimerization, which triggered by the acidic environment of the host cellular endosomal compartment. This results in fusion of the E glycoprotein to the endosomal membrane and uncoating of C protein that releases the viral genome into the cytoplasm. Following this process, translation of the viral proteins, genome replication, and assembly of viral particles occur in the infected cell through manipulation of the host cell machinery. The single-stranded positive sense RNA genome is translated into a single polyprotein, which is subsequently cleaved into individual proteins by the viral and host proteases. Replication of the viral genome occurs in the endoplasmic reticulum-derived membrane, where the positive sense RNA is used to make a newly synthesized negative strand RNA, which serve as a template for synthesis of more single-stranded positive strand RNA. The genomic RNA is subsequently enclosed in the C proteins, forming a nucleocapsid. The nucleocapsid core butts through the ER membrane, therefore acquiring an envelope that contains the major E glycoprotein and the precursor membrane protein PRM. The immature viruses are transported through the Golgi network and undergo proteolytic cleavage of the PRM to M protein by the host protease furin. Mature infectious viruses are then released from the infected cell by exocytosis and are capable of infecting other neighboring cells. As a summary, we have learned about dengue and Zika diseases, where the diseases reside globally, how the viruses were transmitted to individual hosts, and why do these viruses pose a significant health threat to community. We also have learned dengue and Zika viruses, including the virion structures, genome organization, and replication cycle, which are important to understand the biology of these viruses. In the next topic, you will be able to learn about the main mosquito vector that spreads dengue and Zika. We are going to show you facts about Aedes, the mosquito biology, ecology, and behavior. So see you around, guys. Thank you.